Yes, so hopefully it won't be too much of a scary topic, but I'll be telling you a little bit of a story about some of the research that we've recently finalised, and we've just submitted this to Emerging Infectious Diseases. So Mycoplasma genitalium, for those of you that don't know, is becoming an increasingly more important sexually transmitted bacterial infection. It has quite a unique or strange morphology. It's kind of like a flask or a vessel shape with a terminal organelle that it uses to attach to the genital epithelial cells. It's particularly commonly found in cases of non-gonococcal urethritis in both males and females, and it's also becoming increasingly common in pelvic inflammatory disease and also in cervicitis in females. In terms of the prevalence of Mycoplasma genitalium or MGen compared to other STIs, this is actually also on the rise too. So in low risk general populations, we see the prevalence of Mycoplasma genitalium sitting between 1 to 3%. And this is in comparison to other quite commonly or well-known STIs like chlamydia at 4% and Neisseria gonorrhoeae at 0.5%. When we do move to higher risk populations, that's when we do see quite a big increase in the prevalence of Mycoplasma genitalium as well. And so it's particularly prevalent in at-risk populations, and most commonly that includes the young age groups of 16 to 25 years, and also men who have sex with men. And this is really well illustrated in a study that was completed back in 2016 in symptomatic at-risk patients. And what you can see here is that Mycoplasma genitalium is actually incredibly common in these at-risk populations, even more so than chlamydia and gonorrhea. Now, most of what my talk will focus on today is antimicrobial resistance and Mycoplasma genitalium. And so this has become an increasing problem to the point where in late 2019, Mycoplasma genitalium was added to the CDC's antimicrobial resistant threats watch list. So this is the report here. It was released, I think, October or November of 2019. And so, of course, there are urgent threats, things like carbapenem-resistant Acinetobacter, carbapenem-resistant Enterobacteriaceae, some of which is the focus of David Patterson's group, Neisseria gonorrhoeae, which is also a focus of our group. And then if you look down here at the watch list, Mycoplasma genitalium or drug-resistant MGen has now made this list. The reason why this is a big deal is because we've seen the rapid emergence of resistance to two mainstay treatments, and that will be macrolides and quinolones. And we've seen the development of this resistance in less than a decade. So it's really quite a short period of time compared to most other STIs. So just to give you a bit of an idea, so in 2008, we saw the first identification of macrolide resistance in Mycoplasma genitalium. By 2015, the efficacy of macrolides had waned to about 40% for the treatment of Mycoplasma genitalium. And then a study that we've completed where we screened about 400 or 500 M. genitalium positive samples from Pathology Queensland, we found that macrolide resistance is incredibly common in Queensland at about 60% macrolide resistance in those samples. Looking at quinolones, which is the other mainstay treatment that we can use for the treatment of MGen. In 2010, we still had a reported efficacy of 100% for that particular class of antimicrobial. Fast forward to around 2014, the efficacy rate had decreased to 89%. And then studies from Southeast Asia or the Asia Pacific region found that quinolone resistance, particularly in Southeast Queensland, we found that to be around 10% in terms of quinolone resistance. And then a study from around the same time period from Japan found that quinolone resistance is sitting between 10 to 15%. So this is obviously quite concerning because these two drugs are the real mainstay treatments for eradicating Mycoplasma genitalium infections. And so this tells us not only are they becoming much more prevalent in at-risk populations, they're also becoming increasingly difficult to treat with both macrolides and quinolones locally. And so because of this, what we've done, the STI community have done is they've made major changes to the treatment guidelines in Australia, and much of the world has followed this. So in Australia, prior to 2018, macrolides such as azithromycin were the first line treatment for Mycoplasma genitalium. But now, as of March 2018, we now use a sequential therapy. So this includes a 7-day course of doxycycline, followed by either a macrolide, like azithromycin, or a quinolone. And so the choice of the macrolide or the quinolone is based upon the suspicion or the knowledge of antimicrobial resistance. And so for resistance to macrolides, there are commercially available tests from Speedex, who are one of our industry collaborators. And so this can be done within a pathology laboratory, but it's still a little bit more challenging to get quinolone resistance results, because the gold standard for identification of quinolone resistance mutations is still PCR followed by Sanger sequencing. So it's a little bit more difficult to do that in a streamlined laboratory process. Despite the fact that we do have this change in therapy guidelines, and we do have tests, commercial tests that we can use to identify macrolide resistance, local clinicians have still raised concerns over treatment for Mycoplasma genitalium, and this is due to the fact that we're seeing increasing levels of treatment failure with quinolone antimicrobials. And so this is even the case where patients have been shown by our gold standard molecular testing to harbour strains that are susceptible to quinolones. So this really made us think, are we somehow missing quinolone resistant infections? Or alternatively, could the current gold standard Sanger sequencing and PCR be missing low load infections? And that's why we're seeing this treatment failure. So one last thing before I move on to some of the results, I'll talk very, very quickly about how quinolone resistance is mediated in Mycoplasma genitalium. So it's mediated by 4 unique genes. These are the DNA gyrase genes, GY-A and B, and also the DNA topoisomerase genes, PAR-C and PAR-E. And so in our study that we published early 2019 in the Journal of Clinical Microbiology showed that it is mutations in this PAR-C gene in Australia that are very common. And there are a range of different nucleotide mutations represented here. And these are the most common resistance mutations in Queensland. And this is also mirrored in some studies that have been done out of Melbourne as well. So it seems to be that these are the mutations that are associated with resistance locally. So moving on to our study. So we have collected a sample bank of 432 genitalium positive samples from Pathology Queensland over about a 2-year period, possibly a little bit more. And so these samples have been tested by gold standard of PCR and Sanger sequencing of the PAR-C gene. And we've also used additional molecular assays that we've designed within our laboratory in order to try and detect if there are mixtures of quinolane-susceptible and resistant strains within the same patient sample. And so there are, we've completed a PCR that uses allele-specific TACMan probes, and these are to detect the S83 wild type or quinolane-susceptible strain, and the S83I mutation, which is by far the most common quinolone resistance mutation that we see in an Australian population. And then we've also designed some allele-specific PCR primers that specifically detect either the wild type for the S83 and D87 position, and we've also designed PCR primers that will detect the 4 most common resistance mutations, so 3 mutations at position S83 and the most common mutation at position D87. And then finally what we've done is we've also subjected a small number of these samples to PAR-C deep sequencing in order to try and look at the prevalence or the levels of mixtures of these particular alleles within our samples as well. So all 423 samples from our final sample bank were screened using both the TACMan probe and our allele-specific assays, as well as gold standard Sanger sequencing and PCR. Of the 423 samples, 393 of those completely correlated with the results of our gold standard Sanger sequencing, which was excellent. Of these, 341 were considered wild type in that there were no resistance mutations detected, and so therefore they were considered to be quinolane-susceptible. 51 had S83I or D87N mutations, and that indicated that they were quinolane-resistant strains. And we had one very interesting sample that contained both a mixture of the most common quinolane resistance mutation S83I and the S83 wild type allele as well. And this was confirmed in our Sanger sequencing. And so this here is a chromatograph of the quinolane resistance determining region of the PAR-C gene from this sample that I'm talking about. You can see that it's a beautiful, clean chromatograph with the exception of one nucleotide. So at this particular nucleotide mutation here, the predominant allele or nucleotide is a G, which is actually a wild type allele. And then you can see just hiding underneath that is a red peak. And so that peak indicates a mutation from G to T, which correlates with our S83I mutation. And so this was really exciting for us because this told us not only was our assay working and we could detect if there were mixtures there, but in this particular sample, we picked up the mixture, and this was also confirmed by our PAR-C Sanger sequencing. So we were very, very happy with that. But the interesting thing was we also had 30 samples, the remainder of the 423 samples, that had discrepant results between our Sanger sequencing and one or more of our TACMAN and allele-specific PCR assays. So of these, 26 out of those 30 samples indicated the potential presence of mixtures within our PCR assay. But when we looked back at all the chromatographs for each of those samples, we couldn't find any convincing evidence of mixtures like this within our Sanger sequencing. So this, we kind of asked ourselves, are we on the right track here? Were we right? And is Sanger sequencing missing some of these low-level quinolane-resistant infections? So what we did next was we took a subset of 35 samples and we subjected those to PAR-C deep sequencing using the exact same primes that we use for our gold standard PAR-C Sanger sequencing. From those 35 samples, we selected some that had a really nice clear call, so they were either very clear-cut wild type or mutants, and those results correlated across all of the assays that we'd completed so far. And we also subjected some of those 26 suspected mixture samples to Sanger sequencing as well, it was nice for us to see that some of those samples with that clear call also had a beautiful clear call in our deep sequencing. And so 99% or more of the allele present indicated no mixed infections, and that correlated with all of our other assays that said that there was no mixed infection in those samples. Of interest though, for some of those suspected mixed samples, PAR-C deep sequencing did identify mixtures of both wild type and mutant alleles, where our PCR assay had detected mixtures, but where gold standard Sanger sequencing had failed to identify them. And so when we looked at all of the assays that we had completed, we looked across our TACMAN and allele-specific PCR assays and deep sequencing. We found some common mixtures that included the S83 wild type and S83I mutation, S83 wild type mixed with S83I and S83N, which are two common quinolane-resistant mutations. We also saw S83 wild type and S83R, as well as D87 wild type and D87Y mutation, which is a slightly less common resistance mutation, but it's still a resistance mutation nonetheless. And so we were really quite interested to see that there were these mixtures present within our samples. And so the final thing that we wanted to do was really try and look and figure out whether or not Sanger sequencing is robust enough to identify different levels of mixtures. And so what we did was we took some of those beautiful clear-cut wild type and SAD3I samples. We diluted them to the same starting concentration, and then we created artificially mixed samples and then subjected them to gold standard Sanger sequencing and to both of our molecular assays that we had designed in-house. So when they were mixed at a 1-to-1 ratio, we were able to detect it in Sanger sequencing and in both of our PCR assays. However, when we diluted them further, we were only able to identify mixtures at a ratio of 10-to-1 or 1-to-10 for the SAD3I and SAD3 wild type in our SAD3 allele specific assay. So this told us that this particular assay was more sensitive than our TACMan probe assay and also than gold standard Sanger sequencing, which makes sense because this allele specific PCR assay was the one that we identified most mixtures within from our original sample bank, and that was confirmed in many cases by the deep sequencing that we had performed. So in conclusion, we've identified for the first time that there are mixtures of quinolane-resistant and susceptible strains of M. genitalium within the same patient sample. Up to 6% of the 423 samples that we screened may harbour mixtures, and I say up to 6.1% because, like with all studies, there are some limitations. Unfortunately, because of the cost and the sheer number of samples, we weren't able to sample all of our, we weren't able to screen all of our samples by deep sequencing. And so therefore we weren't able to confirm all mixtures by this method. However, the fact that we have found and confirmed the presence of some mixed infections do have very clear implications for the treatment and for resistant screening for Mycoplasma genitalium. So this really for us raises concerns for the potential to detect wild type strains and also to potentially miss resistant subpopulations. We know that this is an issue for other organisms because studies of other organisms like Clostridium difficile have shown that where there are mixed populations of that same organism within that patient's site, it actually becomes far more difficult to treat and eradicate all of that mixed population. So we suspect that we will see similar issues with Mycoplasma genitalium STI infections as well. And not only does this have issues for treatment and testing, it also highlights the complexity of antimicrobial resistance in Mycoplasma genitalium. And it supports the fact that we really do need appropriately designed assays to improve the detection of resistance so that we can therefore enhance patient-directed therapy, make sure that patients are getting on the right therapy, and thus minimize the risk of treatment failure. So just finally, I'd like to thank the people that have been involved in this study, particularly our group for their support, and Kim Lowry for all of her help in screening all of these samples. And I'd also like to thank the people at Pathology Queensland who have helped provide some of the samples and also for providing some of the circa funding that we used to run this study. Thank you.
Fantastic. Well done, Emma. That was really interesting. Questions for Emma? Looks like you might get off the hook, Emma. But I better ask a question. Do you have any clinical data at all on your, do these isolates come with a pedigree so you know whether these patients had a quinolone and what the outcome was, or you don't really have sort of metadata on these samples?
Yeah, unfortunately for a lot of this, we don't necessarily have metadata for this large bank of samples. Mm-hmm. We have been doing a smaller clinical study on a subset of patients where we do have a little bit of this mixtures data. And there's one patient sample that I found just yesterday, actually, where they were treated with azithromycin and moxifloxacin. Mm-hmm. And then, so their pre-treatment sample had no evidence of quinolane resistance, and their post-moxifloxacin treatment had a mixture of both quinolane-susceptible and resistant strains. We don't have any ongoing samples from those patients, so unfortunately we don't know whether or not they were treated with moxifloxacin again or failed treatment. But it's certainly something that we're looking at, potentially getting more clinical data to bolster up these findings.
Fantastic. Well, Emma, thank you. And thank you again, Carlos, for 2 great presentations. I'm very excited the way everyone seems to be thriving anyway. Certainly the people who've given presentations, there's a lot of good stuff going on. So stay safe, everybody.